Click for IB Biology. Here we're looking at the proposed fluid mosaic model of cell membrane structure, which was first published in 1972 by Garth Nicholson and Jonathan Singer, working out of the University of California, San Diego. I remember the publication well, as my biology teacher ran into the room with a copy of the magazine. There's a blank version of this presentation down in the description, a PDF. Quiz has 10 questions with answers, and there's also an AI prompt that you can use to do an individualized quiz of your understanding of this content. So let's take a look at the model presented within the paper. I've also placed a link to the actual original paper in the description, and I think you'll be very surprised at how much of that paper that you're able to comprehend. And so when we look at this model, which was presented by Singer Nicholson, be aware that what they're doing is that they're bringing together lots of observations from different science researchers, and they're working this into a single model. This new model was a hypothesis in 1972 and replaced previous hypotheses because it accounted for all of the observations of cell membranes that were being made at the time, but also brought some new questions about the functionality of the cell membrane. We have the phospholipids in their bilayer with the hydrophilic head molecules here, and the hydrophobic tails in the center of this kind of phospholipid sandwich, which is the bilayer. Let's put that word in there. That's the bilayer structure. And then you'll recognize these features from the other videos that we've done. We have our peripheral proteins, we have our glycoproteins, and we have our channel proteins. We could have added another protein which would have been for active transport, but that would have been a different version of the channel protein. So remember, these can be for facilitated diffusion or for active transport. A feature here that we haven't spoken about yet, but is in the next video, is to do with the role of cholesterol. And that actually relates to the name that the researchers gave to this model, the fluid mosaic membrane. So the mosaic idea is that there are these different component parts embedded within the overall phospholipid structure, but the fluid part relates to the flexibility of the cell membrane. Appreciate that the phosphate heads are not they're not covalently bonded together, and let's take one here, can move in any direction and interchange with each other. What they can't do is move from one layer to the next. That's not allowed, but they can reposition themselves within the side of the membrane on which they are located, and this creates a fluid, flexible, bending capacity to cell membranes, critical, of course, to their function. Another part of the fluidity is that the glycoproteins and the various proteins within it, they can also be moved around and change their position within the cell, and hence the fluidness and the mosaic of the membrane model proposed. So that was 1972. The model, of course, has significantly been upgraded and uprated with the tremendous amount of research which has taken place in more than half a century since then. However, any serious student of biology will get to grips with this model before going on to those more complex aspects of cell membrane structure, some of which are presented in the videos which follow. Some of the more complex and more recent discoveries are to follow. There's also a button here to subscribe to the channel, and indeed the playlist with all the membrane videos included.